So far in biochem, we've talked about how we make DNA and how we transcribe that DNA to make mRNA. Now we're going to use that mRNA as a template to synthesize proteins, which is a process called translation. We're translating the alphabet from the language of nucleic acids to the alphabet of amino acids and polypeptides. Let's start by setting aside mRNA for a moment and let's look at tRNA. tRNA is very important in the translation of proteins or polypeptides. tRNA has a unique cloverleaf structure which you need to be able to identify. Burn that image into your mind. Now, in order to start building a polypeptide, you need to attach an amino acid to each tRNA molecule. We say that the tRNA has been charged with an amino acid, and we call that molecule aminoacyl tRNA. The enzyme that does the charging is called aminoacyl tRNA synthetase, and that's another enzyme worth knowing. And that charging process requires energy, so this uses ATP. A molecule of tRNA has a 3' end and a 5' end. The 3' end is the hydroxyl end, and it always ends with the sequence CCA, cytosine, cytosine, adenine. Now this is the end at which aminoacyl tRNA synthetase works. That's another point that you definitely need to know, so let me say it again. Aminoacyl tRNA synthetase works at the 3' end. So the amino acid is attached to the tRNA molecule at the 3' end so that it can be used to generate the polypeptide. Once the tRNAs are charged and ready to go, you can start translating mRNA and building polypeptides. So where do the mRNA and tRNA come together? Well, they come together in the ribosome. A ribosome is like a little protein factory within the cell. There are tons of ribosomes found on the rough endoplasmic reticulum, which is what makes the rough ER look rough. And then there are also ribosomes found in the cytosol. And it's also worth noting where ribosomes are synthesized. You might think that ribosomes would be synthesized out in the cytosol, since that's where they're going to be working, but they're not. Ribosomal subunits are synthesized in the nucleus and then transported to the cytoplasm. So ribosomes are composed of both protein and RNA, which is called rRNA or ribosomal RNA. So this rRNA is made in the nucleolus, while mRNA and tRNA are synthesized in the nucleoplasm, which is the liquid portion of the nucleus, and that's worth knowing. And each ribosome is made up of two pieces or two subunits. In eukaryotes, like you and me, there's a large 60S ribosomal subunit and a smaller 40S ribosomal subunit, and they come together to form an 80S ribosome. Prokaryotes, like bacteria, have a 50S ribosomal subunit and a 30S subunit that come together to form a 70S ribosome. I want to also briefly mention here that within the 50S ribosomal subunit in prokaryotes, there's a 23S ribosomal RNA molecule. We'll see why that's important later. So for the rest of this discussion, let's focus on prokaryotic protein synthesis so that we can talk about where these different classes of antibiotics interrupt the process. We're going to talk about three distinct phases of translation: initiation, elongation, and termination. Initiation begins with something called initiation factors, or IFs, and they're cleverly named IF1, IF2, IF3, and so on. Initiation factors assist in the assembly of the smaller ribosomal subunit to the first tRNA molecule. So what's the smaller ribosomal subunit? Well, in eukaryotes, that would be the 40S, but we're talking about prokaryotes, so this is the 30S subunit. Now you might remember that the first amino acid in a polypeptide is methionine, so we're always going to start with a methionine tRNA. And if you want to get technical, in prokaryotes it's formal methionine, or F-methionine. So IF2 first binds to the 30S and then to the methionine tRNA. And then when the 50S comes along, it hydrolyzes a GTP on the initiation factor, which releases energy, and that allows the 50S to attach to the 30S, so that now you have a complete 70S prokaryotic ribosome. There are three sites on the ribosome complex where the tRNA can bind. The A site is where the incoming aminoacyl tRNA binds, A for aminoacyl. The P site is where the polypeptide tRNA binds. This is where the growing polypeptide chain is found, so P for polypeptide. And the E site is where the free tRNA is located, right before it exits the ribosome, so E for exit. Now, the first tRNA, that F-methionine tRNA, binds at the P site rather than the A site, but all the subsequent aminoacyl tRNAs bind at the A site. So that's initiation. Now we're ready to move on to the next phase of the protein translation, which is elongation. This is where we start putting these amino acids together in a long chain, which is our polypeptide. The incoming charged aminoacyl tRNA binds the ribosome at which site? The A site. Remember, A for aminoacyl. And this binding requires not initiation factors, but elongation factors, or EFs. So the initiation factors help to put the ribosome together. Now elongation factors help the incoming tRNA to bind to the A site. And again, this uses a little bit of energy from GTP. So now that you have these two tRNAs side by side, one on the P site and one on the A site, There's a part of the large ribosomal subunit, the 50S, that has what we call peptidyl-transferase activity, which transfers the amino acid from the P site onto the amino acid at the A site. In prokaryotes, this peptidyl-transferase activity is found in that 23S ribosomal RNA, which is a type of ribozyme, an RNA enzyme, within the 50S subunit. So the 23S rRNA transfers the peptide from the P site over to the amino acid on the tRNA in the A site. So in this case, where we're just getting started, it transfers the methionine initiator onto whatever comes next, which just happens to be lysine. The next little step, which is still part of elongation, is called translocation, which is where the ribosome complex moves down the mRNA, just 3 nucleotides, and this moves the tRNA with the growing polypeptide chain from the A site to the P site so that the process can repeat. This process also moves the newly uncharged tRNA from the P site over to the E site, remember E for exit. And that tRNA is going to get kicked out, and it can go back and be recharged by aminoacyl tRNA synthetase. And there are a couple of players involved in translocation that you need to know for your test. Translocation requires elongation factor G in prokaryotes and elongation factor 2 in eukaryotes. Make sure you know those. That's actually clinically important because there are a couple of bacterial toxins that work by inhibiting EF2 in host cells, diphtheria toxin and also exotoxin A, which is produced by Pseudomonas. So elongation just keeps going and going and going as long as there's mRNA code to translate. The new aminoacyl tRNA comes in at the A site, the 23S rRNA peptidyl-transferase moves the polypeptide chain from the tRNA in the P site over to the tRNA in the A site, and then the whole thing slides over one spot and starts over. As long as the mRNA codon at the A site is valid, the ribosome is going to keep going. But what happens when the codon at the A site is a stop codon? Remember, the 3 RNA stop codons are UGA, UAA, and UAG. And if any one of those 3 codons turns up, that's a signal to the ribosome that we've reached the end of the line and it's time to terminate. So this is the final step in protein translation, which is called termination. There isn't really much to know about termination. Basically, those 3 stop codons don't match up to a tRNA, so there's no new tRNA coming into the A site. Instead, something called a release factor binds to the mRNA, and it hydrolyzes GTP again, so this is yet another step that requires energy, and then the new polypeptide is released from the tRNA at the P site. And then the ribosomal subunits and the tRNA all separate from one another, and they can be reused.